your faces, although I can't. And oh, it's also now streaming on YouTube. Wow, great. Okay, it looks like we're up and running. So hi, good afternoon. Welcome to the Ecology, Evolution, and Conservation Biology talk today. Today we are joined by Jessica Ware. So if everyone could give a warm virtual welcome to today's speaker, leave time for applause. All right. Okay, I'll stop making nerdy jokes really soon, I promise. So just to get everyone started and get to know our speaker. So Dr. Ware is zooming in from New York City, where she is an associate curator in invertebrate zoology at the American Museum of Natural History. She specializes on the evolution of behavioral and physiological adaptations in dragonflies, damselflies, termites, cockroaches, mantises, so all of the creepy, crawly good stuff. But more importantly than all of that, Jessica is Canadian. She got her BSc from the University of British Columbia, a PhD from Rutgers, New Brunswick, and that's pretty ironic because before heading to AMNH, where she's at now, she was an associate professor over at Rutgers in Newark. She's president of the Worldwide Dragonfly Association, VP of the Entomological Society of America. She was awarded a P-Case medal from the US government for her work on insect evolution, and I hear tell that she's a pretty great punk rocker to boot. Today, we're excited to welcome Dr. Ware and learn about the first transcriptome-based and AHE-based phylogenetic reconstructions of Odonata, representing nearly all of the order's families. So thank you for coming today, Jessica. Hopefully you feel at home here in Beaver Nation.
Thanks so much for the introduction, and it is nice to be visiting a department with fellow twins, with other Canadians, with all of you. So thank you for having me. So as was mentioned, these are the groups that I mostly work on, and I'm going to be talking to you about dragonflies and damselflies and some of the work that we've done on Dictyoptera. And I would just note that like many of us who go into the field of entomology, I'm really a field biologist at heart. I mean, I'm interested in understanding the evolutionary history of insects because when I go to the field, whether it's to sample things that I specifically need for a phylogeny, or if I'm in the field doing behavioral observations, that's when you really start noticing some of the amazing variety of insects that are out there. And that's kind of what inspires me to do the evolutionary biology work that I do. I didn't really go into it to pipette clear colorless solution from one tube to the next. It was really this, just the sheer biodiversity, the immense number of species of insects that are out there that really dwarf all of the other organisms that are on Earth. This is really what got me kind of interested in creating a research program to try and unravel why some groups are really species rich and other groups are really species poor. And certainly when we look across the orders of insects, what we notice, and this is something that I noticed even as a child, although I wouldn't have had the words to kind of articulate this, but I would have said at that time, wow, there's a lot of different types of insects. Now I could say, there's extreme heterogeneity in phenotype and genotype, in size, form, function, and all of this insect kind of variety is basically what we study using phylogenies. So I'm interested in understanding how these different orders of insects are related to each other, when insects first diversified and subsequent diversifications that have taken place. And then ultimately, which is kind of a life goal, I guess, what we want to find out are some of the drivers, the why, why are some groups so speciose, and we see a lot of variation in species richness across insect groups. So the groups that I mostly work on are the Bladodea, which are cockroaches and termites, and then the Odonata, which are the dragonflies and damselflies. And I'm mostly going to focus on odonates today, but I'll mention a little bit about termites and cockroaches. So I'm going to just briefly kind of talk a little bit about the phylogenetic work that we've done, some of the population genetic work that we've done. And then I thought it would be, nice to kind of highlight a little bit about why we're doing the work that we're doing to diversify entomology as a discipline. So it's a diverse group of organisms. We would like to have some diversity in our field as well. So ultimately, kind of what we're doing in these types of studies are making trees and then using fossils to calibrate the trees. And that's kind of getting at this how are insects related to each other question, and then the when question, when are these different insect groups first diversifying? And when I was at UBC, I went to UBC, as Nat mentioned, for my undergrad, and Jeff Scudder was the person who taught me entomology. And I can remember him distinctly saying there were these key innovations, after which we see rapid radiations of kind of insect taxa. And certainly, if you think about the key innovations that may have been some of the drivers for key diversification events, in terms of Platodia, what we're interested in really is the evolution of social behavior. But in terms of odonates, what we're really interested in, what we talk a lot about in this field, is flight behavior. So this is a phylogeny that colleagues and I reconstructed in 2014 using transcriptomes. It's kind of an insect tree of life. And I'm going to use this phylogeny kind of as a map, I guess, or a guide as we move through to discuss a couple of the different orders that we're going to discuss today. So odonates are located in this part of the topology. So if we kind of zoom in here, we reconstructed them in this phylogeny as sister to the mayflies. And so when we think about the number of species of mammals, there's about the same number of species of odonates. So it's not like beetles or Trichoptera or Lepidoptera where we have hundreds of thousands of taxa. There's 6,300 species of odonates, and this order is divided up into 3 suborders. The dragonflies, which are the Anisoptra, there's around 3,000 species of those. The damselflies, which are the zygotra, there's around 3,000 species of those. And then there's this strange kind of enigmatic taxon, exists today as a single genus, Epiphlebia, that's found in the Himalayas, China and Japan. It has kind of an abdomen that's stocky like a dragonfly, but petiolate wings like a damselfly. And just like when we look across insects and we see amazing variety, so too when we look across the order Odonata, do we see extreme variation in size, in color. Dragonflies are perhaps most attractive to humans because of their remarkable coloration. And they have a couple of different ways that they have color, both in the wings and in the body, that are interesting, that I'm happy to talk about later if there's time. So this attractiveness, this wow factor of how beautiful they are, has led humans to use these for everything from Tiffany lamps to tattoos to bowls to jewelry, and it's always usually the adults that are the ones that folks use for decoration. These are aquatic insects, though. So both for Anisotra, zygotra, Anisopra, for all of them, they're voracious predators that dwell in fresh water for their nymphal or larval stages. Some of them have very short durations. They are in the water 4 to 6 weeks. Others develop over 4 to 5 years in fresh water. And just like the adults, they consume other insects, they consume fly larvae and what have you, they consume each other, but they also are able to consume vertebrates. So they routinely take fish and tadpoles as their prey. And so when we look at the kind of earliest branching lineages within the pterygoda or the winged insects, one of the things that people are interested in terms of the dragonfly story or the play that involves dragonflies is if they were the first things to take to the skies or whether or not they perhaps share a common ancestor with mayflies, which form a group called the Paleoptera, which is sister to the Neoptera. There's this hypothesis that dragonflies were the first, the earliest branching lineage. And then there's this other hypothesis that perhaps mayflies were the earliest branching lineage. And I and many dragonfly people, I think, kind of favor the second hypothesis, the Termitaria hypothesis, which has dragonflies as sister to mayflies as sister to the Neoptera. And part of the reason for this is that they have indirect sperm transfer. So basal hexapods also have indirect sperm transfer. They will make small packets of sperm called a spermatophore, and males will bring the female over to the spermatophore, and she will then take the sperm in this little packet. Dragonflies and damselflies don't do that, but they have something that they think perhaps could have evolved from that spermatophore presentation that we see in basal hexapods. It's called indirect sperm transfer, where males have a sperm that is ejaculated from their primary penis at the tip of their abdomen. A secondary set of male genitalia at the base of the abdomen, and the secondary genitalia, they ejaculate into that secondary genitalia, and then they use that for sperm transfer. So instead of the sperm going directly into the female, it goes into this other vesicle, and then that is where the sperm is pumped into the female. So they also can mate multiple times, and males want to ensure paternity. So this is actually what the secondary genitalia looks like just under a microscope, but it actually is even more elaborate than that. It has these long hooks and claspers, and some things that almost look finger-like projections that males use for sperm displacement, either scraping out the previous male sperm or packing deep in the sperm storage organ the previous male sperm, we think to try and ensure paternity. So when you see males and females together, they form this feature called the copulatory wheel, this kind of heart-shaped union as they're reproducing. But the majority of the time when they're together is actually sperm displacement, and the actual sperm transfer happens very quickly. And again, with this indirect sperm transfer mechanism. So people have been interested in kind of understanding how these different groups of dragonflies and damselflies are related to each other for some time. And over the last couple of hundred of years, people have largely looked at perhaps the most visible feature, which is wing venation. So most of the early odonate systematic work that was done in taxonomic works was really based on variation in the pattern of wing venation. And there are some differences as you look across families in terms of the relative position of various veins. But what research has showed us is that there's the arrangement of veins, the density of veins, whether there's very densely packed veins or sparse regions of veins, that actually is correlated with certain styles of flight, because very dense venation makes the wing very stiff. And areas of the wing where there's less wing venation, the wing can be more floppy. And so depending on whether you have long distance style flight, short distance style of flight, maneuverability in and amongst vegetation, that seems to be a better predictor of the wing venation. And wing venation is not necessarily a good reflection of evolutionary history. So many of the groups that were described, or the arrangements, the relative kind of arrangements of families that were based on wing venation, haven't really found to be upheld with molecular evidence. So a lot of the work that's been done in terms of trying to redefine the taxonomy within odonates has been in the Zygotra. And this is in part just because it's chaotic, right? So in 2013, colleagues and we wrote a paper kind of defining the taxonomic groups within odonates. And at that time, I think there was, I think we thought maybe there was 13 or 14 families of Zygotra. I forget how many we thought there was. Anyways, now there's 50. That have been described in some form or other. So a lot of work is happening in the Zygotra. We've done some work on some of the Zygopteran families. But today I'm going to really focus on the Anisoptra. And in the Anisoptra, the biggest questions that we have really have to do with relationships among three main taxonomic groups. So unlike for the Zygotra, there's far fewer families that have been defined within the dragonflies. There's only 10 families that have been defined, but there are some nodes that have been notoriously difficult to try and resolve. And the interfamiliar relationships between these three groups are just kind of a mess. So the first is the Gomphidae, or the clubtails. The second is the Pedaluridae, or the petaltails. And then the third taxonomic group is actually a superfamily called the Libelluloidea. And I've got some examples of the 4 families that comprise the superfamily kind of colored here in blue. So just to kind of walk you through what these families look like, the group that most think was the earliest branching lineage within the Anisoptra are the Aeschnidae. There's around 450 species of these, and this group includes long-distance migrants like Anax junius, which we know to be a long-distance migrant. They're large, they're really colorful, they can kind of migrate granules of pigment in their epithelial cells to make themselves lighter or darker in color. They're kind of a remarkable taxonomic group. The Gomphidae, or the clubtails, are a very speciose group. So it's the second most species-rich group within the Anisoptra. There's over 900 species. And these clubtails have a reduction in the ovipositor, or their egg-laying apparatus. And instead of laying their eggs the way that damselflies and most other dragonflies do, they actually just squirt out their eggs in a clump and tap their abdomen on the surface of the water to disperse their eggs. The Petaliuridae are a really species poor group by comparison. There's only 10 species that are found, and the majority of the species are actually in Australia, with one in South America and 2 in North America. And here's a Sasquatch joke for you. So this taxon was actually studied by someone. His name's Perry Turner. He did a PhD on this taxon at Berkeley. And when he was doing this work, he spent a lot of time in Oregon and the Pacific Northwest, sampling for petaliurids, for Ctenipteryx. And in doing so, he became convinced that they were the primary diet of Sasquatch. And so in his thesis, he wrote, and actually handed this in this thesis, he wrote, Sasquatch are rare because their prey item is Ctenipteryx, which is rare in the habitat. So even though I joke about him, he was a remarkable biological illustrator. This is actually one of the plates from his PhD thesis. He ended up disappearing into the Pacific Northwest. And there's a whole story that goes on with what happened to his artifacts, which I can tell you perhaps after if there's time. So the most species-rich group is this superfamily, Libelluloidea, which comprises these 4 families. And there's over 1,500 species of these. These also have a reduced ovipositor. So they have a reduction in this apparatus for laying eggs, and they just squirt out their eggs in a clump. So what we did was we wanted to try and use molecular data to really broadly sample across the Zygotra and the Anisoptra. And we did this with a couple of different types of data. So first what we did was we used anchored hybrid enrichment to sequence close to 500 loci for 136 species across the Anisoptra and the Zygotra. And in doing so, of course, I told you Zygotra, it's chaotic in terms of their systematic. So in doing so, we actually found some new families within the Zygotra that were proposed, and 2 more were reinstated. A couple of the groups that have been well studied in Zygotra by ecologists, by people who are not evolutionary biologists, were things like the demoiselles or the ruby spot. These have been commonly used in sexual selection studies. Well, it turns out that the superfamily to which they belong is actually paraphyletic, the Clopterigoidea. I mentioned before, people were really interested in what was the first thing to take to the skies. We know that there were these other things called griffin flies, which are not crown odonates or not modern odonates, and these were abundant in the Carboniferous period. But in terms of what was the first thing that was actually within crown odonata, some people suggested it was Damselfly-like, some people suggested it was Anisoptra-like, and one hypothesis that people had was perhaps that the earliest branching lineage was a taxon that still exists today. Hemiphobiamirabilis is a damselfly that exists only in these 6 really fragmented populations in southern Victoria and northern Tasmania in Australia. And based on morphology of both the nymph and the adult, as well as some of the reproductive behavior of the female, she kind of arches her abdomen over her back and then just kind of throws her eggs across the marshy habitats, boggy habitats where she lays her eggs. People thought perhaps this actually would be sister to all of the other odonata. So we were fortunate enough to get some samples of this taxon, and we recover it in this first branching clade within our topology as kind of sister to the Lestoidea, right here. That third suborder, the Anisozygotra, the one that's only found in the Himalayas, China, and Japan, that has wings like a damselfly but a body like a dragonfly, we recover it as sister to the Anisoptra. This wasn't surprising, and I think there's a lot of people that really support the idea of just having that be a single suborder with Anisozygotra and Anisoptra together. The name is Epiprocta, and with that just being the suborder. So one of the questions that we had was, for the 2 most speciose group, which are the Gomphidae and the Libelluloidea, these are both the ones that have this reduction in the ovipositor. So females have lost their gonapophyses, and instead they just squirt out their eggs in a clump. We wanted to see whether or not this phylogeny could help us answer this question: what's the ancestral egg-laying strategy for dragonflies? And one of the things that we think may be driving a reduction in the ovipositor is the speed. So when they squirt out their eggs in a clump, they can actually just, lay their eggs within a matter of seconds, whereas all damselflies and the other dragonfly families, they have an ovipositor that's kind of like a little knife, and they cut a hole in plant material and put their eggs in one at a time. And that leaves them really vulnerable because it's time consuming. It leaves them really vulnerable to taxa like the ones that are shown here. So fish will jump out of the water to catch dragonflies, birds, lizards, frogs. They all impose some pretty severe predation pressure on dragonflies, especially females when they're laying eggs. But to get at this question of what the ancestral egg-laying strategy is, we really need to have a good topology, a really good understanding of the relative familial positions within the Anisoptra. And the reason why that's tricky is because to date, people really haven't been sure where Gomphidae and Libelluloidea fall. So if they're sister taxa, that means that perhaps the reduction in the ovipositor, which is called exophytic egg-laying, without plant egg-laying, that would have just perhaps been most parsimoniously explained as having evolved once. But if Gomphidae are sister to the Pedaluridae, and the Pedaluridae are the kind of Sasquatch food, if they're sister to the Pedaluridae, that would suggest that this reduction in the ovipositor occurred twice. And so we really need to have a good resolved phylogeny so that we can answer this question. So with our anchored hybrid enrichment data, again, we had close to 500 loci. We found that Pedalurids and Gomphids are sister taxa. So we find strong support with bootstrap support, strong support with posterior probability. We find strong support if we look at quartet sampling. And again, this sister group relationship suggests that this reduction in the ovipositor in the Gomphidae and in the Libelluloidea would have occurred twice. So this exophytic egg laying eggs without plant material by just squirting out your eggs in a clump seems to have evolved twice. But I mentioned before that we tried a couple of different types of data to get at this question. And so with my postdoc, Manpreet Kohli, we also, and our colleagues at OneKite, we also sequenced transcriptomic data for 105 odonates, slightly different taxon sampling, but same breadth across the damselflies and the dragonflies. And what we find with these data is the same thing. So we find Pedaluridae and Gomphidae as sister taxa with strong support. We also ran in this analysis 4-cluster likelihood mapping, which is a quartet sampling approach. And what we find is it basically kind of sorts the trees that you get into different piles depending on which hypothesis they support. And this actually, this part of the triangle that says 88%, the majority of the results kind of actually supported Gomphidae as sister to the Libelluloidea. But when we looked more closely into this, it seems like some genes are very strongly supporting that relationship and other genes are very strongly supporting Pedaluridae and Gomphidae. So this is another, I guess, unsolved mystery. It's nice because it involves Sasquatches, so I like saying unsolved mystery. It's another unsolved mystery in the Anisopteran tree. We did spend a lot of time trying to estimate divergence times for this group, again, because we really want to get at the driver, perhaps, of this reduction in the ovipositor. And we know that some of these vertebrate predators impose such extreme predation pressure on these dragonflies. And so we tried a couple of different calibration schema. And what we found was that in large part, the majority of the diversification that occurs in the groups that have the reduction in the ovipositor is during this time period here when we see the rise of modern birds. It's possible that this provides a little bit of extra evidence that predation pressure may have driven this kind of switch to a very fast egg-laying strategy, but we have a lot more work to do. I will just note briefly that the way that the ovipositor is reduced in the Gomphids and in the Libelluloids are slightly different in terms of the morphology and musculature attachments. So more broadly, thinking beyond just the reproductive biology, thinking beyond the questions of what's happening within these specific families, for odonates in general, we kind of want to know why there's such diversity and why certain groups are kind of highly diverse and other groups are not, like the Pedalurids with only 10 species. And so one of the key answer seems to be color. The groups that have the most variety of color seem to also be the most speciose groups, but also dispersal ability. And so we've started to look at the dragonflies that have the widest, that are the best dispersers, kind of one by one, to really better understand their biology so that we can ultimately get at this question of whether or not dispersal capability is a driver of diversity in this group. So I thought I would just mention some work that we've done on one of these taxa, which is Pantella flavescens, the global wanderer, it's called, or the wandering glider. It's a libellulid that's found somewhere in this part of the tree. This is a family that has been notoriously difficult to kind of resolve the intergeneric relationships. What we know about Pantella is that it is a cosmopolitan global migrant. So regardless of whether or not we've sampled them in Australia or Guyana or Canada or Texas or Japan, it's almost always the most common thing that's collected, and it is one of the most abundant dragonflies that people have in museums and other collections. And one thing that's kind of remarkable about it is that this genus is pretty small. There's only 2 species in it. So there's Pantella flavescens, which is a cosmopolitan dragonfly. And then there's Pantella hymenea, the sister taxon, which is only found from Canada to Argentina. And they look very similar, only Pantella hymenea has this kind of little brown spot at the base of its wings. So what we know about Pantella flavescens, we don't know as much about hymenea, but what we know about Pantella flavescens is that it has the characteristics of a long distance migrant. So it has very rapid development. It often takes advantage of weather. So it takes advantage of temporary and transient bodies of water that kind of appear after big rains. And so to take advantage of temporary, transient, ephemeral water sources, it kind of is logical that they have a very rapid development time of about 5 to 6 weeks. This is in comparison to some other dragonflies, like I mentioned, that can, be 4 or 5 years in freshwater developing. They have some unique morphological adaptations. They actually have this section of their wing, which I've kind of colored here in red, are the anal veins. And there's an expansion in the anal vein region. And many of the dragonflies, this is not unusual. There are some other dragonflies that have an expansion in the anal vein region. And all of the dragonflies that have this tend to do kind of gliding style flight. We think it kind of increases surface area and maybe decreases energy expenditure. And then they take advantage of existing wind patterns. So they take advantage of the intertropical convergence zone, which is like a series of winds that kind of go around the equator. And they have this behavior when wind passes over the surface of their wings, there are these small little hairs that trigger a reflex for them to pick up their feet, and then they get carried by the wind. One thing that's interesting, though, is that anecdotal evidence, it's anecdotal, mind you, but anecdotal evidence from people who have traveled to remote islands like Tonga or Easter Island, they noted that there was a different behavior. So when wind passed over the surface of the Pantella on Easter Island, colleagues noted that they actually crouched and got lower to the ground. And the authors of this paper argued perhaps that was to avoid being blown out to sea. So these dragonflies often traveled in huge swarms and then pass out, fly out over open ocean. But freshwater is their zone, right? So they don't do well with saltwater. They actually will die in saltwater. So what we think happens is they just basically fly until their fat stores are depleted, and then they just drop into the ocean dead. So the majority of individuals that leave India, for example, cross the Indian Ocean to go to East Africa, the majority of individuals don't actually survive that migratory route. So it would make sense that if you're on a remote island, one could imagine how selection could favor individuals that had a modified behavior to not be blown out to sea. So we wanted to get at this question, if you have something that's globally distributed, what is a population in this scenario if it's globally migratory, truly panmictic? So we sequenced individuals from both of the species in this genus. We did this because there actually was some question whether hymenea was just a different phenotypic variant from flavescens, but we do recover them as 2 separate clades. And when we sequenced the cytochrome oxidase 1, a gene which we chose because it would allow us to also kind of harvest information from NCBI and public databases. Like I said, it's a really commonly collected dragonfly. So people have sequenced the barcode gene for Pentella from all over the world. So we were able to include those sequences. We were able to kind of assemble them into this data set of over 700 sequences, close to 800 sequences from the countries that are colored here in these different colors. And what we found when we did kind of genetic analyses is that there was really high evidence of gene flow kind of across the ranges, across the geographic ranges where we sampled them. And these figures here that I'm showing are called haplotype networks. And haplotype networks basically show clustering of genetic patterns. So each circle indicates a different kind of genetic pattern. The size of the circle tells you something about the number of individuals that share that genetic pattern. And these little tick marks on the lines indicate the number of nucleotide differences between one pattern and the next pattern. And what you'll note is that the majority of individuals from our data set share one genetic pattern, regardless of whether they were from India or Indonesia or Guyana or Brazil or Texas. They all kind of mainly share this one main genetic pattern. And we didn't see a lot of variation. We thought maybe there would be kind of a hemispheric divide between Western, Eastern, Northern, Southern Hemisphere, and we don't see that. A friend of mine collected one that landed on the edge of this hot tub that she was in when she was on a cruise in the Indian Ocean with her mother. And she collected it and gave it to me, and I sampled it, and it falls right here with this same main genetic pattern that's the most commonly found. So another way that you can kind of look at whether or not something is sharing genes kind of across a long distance is to actually look at whether or not the individuals that you collect as adults actually emerged from water in that same location. So like I mentioned, all dragonflies and damselflies have freshwater nymphs, so they develop in freshwater and then emerge as adults. And the wings actually develop while they're in freshwater underneath the larval skin. So the hydrogen that's in the wings is actually incorporated from the water, from the H2O in the water. And so you can look at the weight of hydrogen in wings, and it turns out hydrogen actually varies in weight along a latitudinal and longitudinal gradient. You can look at the weight of hydrogen in the wings by combusting them, collecting the hydrogen and weighing it. And it can tell you something about whether or not the individual you collected it, say in Senegal, whether or not it had been a nymph in Senegal or whether it had been a nymph in Toronto, and you collected it in Senegal. So we did this analysis, this isotope analysis, with a subset of our data, a subset of our specimens. And what you get are kind of these heat maps that look sort of like this, where different colors kind of indicate the range of predicted hydrogen weights that would be found for a given region. And then you can compare it to the weight that was recovered for the wings that you basically burnt, that you combusted, that you collected from that particular region. And what we found was that the majority of individuals that we sampled, and we sampled from these different countries that are shown here with different colors for each country, the majority of individuals had, different, significantly different hydrogen weights than what would be predicted if they had been born in the country or in the region where we sampled them. So the individuals that kind of fall above this gray line are truly migrants. So they were individuals that were born from water, pond or lake or what have you, in a region different from the area where we collected the adults. There were a couple of exceptions where there were individuals, particularly individuals that we sampled in the Andes, that had signatures that suggested that perhaps they were local. But you can look at a dragonfly wing and you can see how tattered it is, and that tells you something about the age of a dragonfly. The longer an individual has been around, they're around for a couple of months, a year as adults, the more tattered and banged up their wings get. Teneral dragonflies, which are dragonflies that have recently emerged from water, by contrast, have kind of shinier wings with very few tatters on them. And the individuals that we had sampled from the Andes, it looked like they were perhaps recently emerged individuals. Not old individuals, but ones that perhaps were either teneral or were relatively recently emerged. So it's possible that maybe these would have become migratory, but these were from museum samples. The individual, the human that caught them, a day or two after they had emerged. We don't know. So what we'd like to do, this kind of has inspired us to do a time series and kind of do the same analysis again, estimating whether or not we have local or migrant taxa, but kind of across seasons to see if we can kind of parse out that data. And my graduate student, I'll just mention Stephanie Mofflin-Mills, has been really interested in another anecdotal, mind you, but an anecdotal observation that some colleagues made. So some colleagues noted that on Easter Island, on Tonga Island, on a few of the smaller islands, remote islands, they thought that perhaps the size and shape of the Pantala wings seemed different in some way. So Stephanie's using an automatic feature extraction software that we developed in my lab called Odomatic, like automatic Odonata feature extraction, she sampled wings kind of across a range of Pantala. I think she's, scanned 400 or so individuals to really test whether or not there are significant differences in the morphology of the kind of size, shape of wings and the density of wing venation for these island and mainland populations. In terms of the other species, Hymenea, we really don't know almost anything about it. So we're starting to work on this taxon, but it's much more rare in collections. And we think maybe it's a north-south migration that's occurring in this taxon, but more on that soon. So that's kind of all I'm going to say for this part of the tree. And now we'll move to the Polynyatra. And within the Polynyatra are the Dictyoptera, which are the termites, cockroaches, and praying mantises. So like I mentioned, I wouldn't say that I'm necessarily super fascinated in the other members of the Dictyoptera other than the termites, but since termites are cockroaches now, I guess I'll say we'll talk about cockroaches. So I've been working with one kite and my former graduate student, Dominic Evangelista, on trying to, for the first time, really get some strong resolution in terms of how the different families and superfamilies within Blattodea, how their relationships are. And what we know from transcriptomic data is, as has been found with other data, termites are nested well within the Blattodea tree of life. And so what we've done is we've tried to, just like we've done for dragonflies, use a variety of data from transcriptomic to anchored hybrid enrichment data to really test some of these relationships that we're finding among the families. We do find that we're converging on the true tree of cockroaches with a heavy emphasis on the Blabberoidea, which is, where the majority of the sampling has been done. But what's interesting is that just like for the dragonflies, we sequenced, 500 loci for the dragonflies. We sequenced 200 and some odd loci for the cockroaches. But it turns out there's actually just a subset of those loci that are the most informative. So we recently published a paper just this past year where we kind of did subsampling of anchored hybrid enrichment loci so that we could make recommendations on the cheapest and most efficient pipelines that would still resolve, the cockroach tree of life, the dragonfly tree of life, and what have you. For termites, what we mostly have been interested in is diet-driven diversification. So we know that, termites eat wood. That's something that is commonly discussed, but termites also eat soil. They also eat bits of twigs and grass. And there seems to be a switch between what they call the lower termites and the higher termites in terms of the complexity of their diets. And this is related to the endosymbionts that live in their hindgut. And so my graduate student, Megan Wilson, her whole thesis really is looking at diet-driven diversification and mandibular morphology changes that are associated with shifts in diet. But I'll just quickly do a foray into a termite of interest to us, which is actually unrelated to this question of diet-driven diversification. But we happen to be in Guyana sampling termites for this project about diet-driven diversification. And we came across these termites that had these kind of pads on their abdomen, these kind of six-packs. And they also were creosote-like. They kind of exploded with soil and other food that they had ingested. It turns out this is actually a fungus. This is a fungal ectoparasite called Termitaria, is a genus of fungus. And this kind of led us into this foray to look at the different types of fungi that infect termites. People know a lot about the fungi that infect ants, but it turns out, termites too are very susceptible. So we recently just published a paper. It just came out in Annals of the ESA, describing all of the different fungi that infect termites. And Megan is a terrific biological illustrator. So in this paper, she actually, in great detail, the minutiae are there, kind of describe the life history strategies of both the fungus and the termites and the modes of infection. So just to kind of conclude about what we found in our research so far, I think we are, for both cockroaches and termites, and dragonflies and damselflies, we're kind of starting to converge on what we think the true tree of life is, using anchored hybrid enrichment data, transcriptomic data, morphological data, looking at population questions. That's allowing us to kind of do deep dives into specific taxa that might hold clues about some of these drivers of diversification that we're interested in. And so the question, kind of is in entomology in general, but certainly in our research program, is what to do next. Okay, we've got our phylogenies. Well, the thing that we're really interested in is now working on diversifying our field. I talked briefly about the past, about kind of a hero of mine who is Margaret Collins. Dr. Margaret Collins is a termite biologist, or was a termite biologist, and I'm kind of fascinated by her life and her journey, which in some ways is very similar to the experience that Black women in entomology are experiencing today. So she was fortunate that her parents, really encouraged her to go into education. Her father worked for George Washington Carver. She was at this in this town full of Black academics called Institute in West Virginia, and she was really encouraged to do a bachelor's degree in 1943. And then, remarkably, she went. I mean, I find this to be remarkable given the experience I had when I was applying to grad school. Remarkable. She was really encouraged as a Black woman in entomology to go for a PhD. And she became the first Black woman to receive a PhD in entomology. I think it was in 1950. She worked with Alfred Emerson, who was at the time at the University of Chicago, who had a really extensive research program in Guyana. And so Margaret worked on Guyanese termites. As an aside, Alfred didn't let her ever go on any of the fieldwork trips. He didn't believe that women were designed for fieldwork. He didn't think that they should go to the field. So all of the work that she did on these termites, she actually just had to do looking at specimens that he had brought back with him. She ended up going and working for a historically Black college as a termite biologist, and she was becoming well known as a termite biologist. And so she was invited by the University of Florida to talk on her research. And when it became public knowledge that she was going to be giving this talk, this was the White School in Florida. University of Florida received a bomb threat. And the school was threatened of, with being blown up if this Black woman was allowed to come and speak on her research on termites. And so her talk was canceled. And around this time, there was a few crossbreedings that had taken place that led students at her school to start a bus boycott. And so she became a driver for the bus boycott. So people were boycotting the bus, but they still needed to get to work and they had to carpool to go get groceries and what have you. So she became a driver for this bus boycott, and still published a bunch of papers on termites while raising 2 kids on her own, I might add. She ended up going to become a professor at Howard University, and she did a bunch of the early work on Anisotra's defensive secretions. So Anisotra are a group of termite that have these kind of snaz-like apparatus. Their fontanelle is modified so they can squirt out this gooey-like secretion that kind of paint on with these little hairs at the tip of the nasus. And then eventually she ended up deciding to go to the Smithsonian, where she worked as an unpaid research associate for the remainder of her life, which allowed her to not have to teach, and she could just kind of focus on doing her termite work. And so in 1977, she established this Guyanese field station, which she called the Alfred Emerson Field Station on termites. And she had this really elaborate program studying Guyanese termites. And she ended up dying in the field, doing what she loved, sampling in the Caribbean in 1996. And I mentioned Margaret because I feel like, although I mean, certainly some people have certainly heard of her. She was a prolific scientist. some of the things that she went through are actually not that, dissimilar from what I hear some of the colleagues that I have, are experiencing in today's climate. Being the first Black woman to receive a PhD in 1950, I actually found out writing a paper this summer that I was the first Black woman, first Black person, period, that had received a PhD at Rutgers in entomology in 2008. When I got that degree, I had no idea, but I was the first Black person to have gotten that degree. Now I'm a curator at the American Museum, and I became the first African-American woman curator that they've ever had in 150 years. So there's still a lot of firsts. Margaret made that first, she was the first Black woman to get a PhD in entomology, but there's still a lot of firsts that are still happening and that still need to happen. And as an aside, I'm in charge of Alfred Emerson's collection, which was donated to the American Museum. There's a photo of my students and I with this portrait of him next to his card catalog. We do a lot of our work in Guyana, and I like the idea that my mostly female graduate students and I are going to the field all the time, contrary to what Emerson would have thought was appropriate. And I'm sure Margaret would think that was kind of cool if she was able to know that. So what we know from the current situation is that the numbers are pretty low in terms of diversity across ecology and evolutionary biology. And when we look specifically at entomology, we wanted to know if we spent some time this summer trying to see whether or not these numbers that we see reported for ecology and evolution were broadly true across entomology. And so some colleagues and I wrote a series of papers this summer, and I'll just briefly mention this one, which you published in the magazine American Entomologist. We kind of did a few things in this paper, including surveying departments to see when the firsts of their department were, which is when I found out that I had been the first Black person to get a degree at Rutgers. But one of the things that we did in this study was to kind of really delve into the data that's provided by NIH and NSF and some of these federal scientific reporting agencies. They tend to group entomology and parasitology together, but I'll just briefly mention that what we find is that compared to some other disciplines like psychology, which seems to have some, a bit more proportional representation compared to what you would expect from the human population, we don't find that for entomology and parasitology. So we find that there's extreme underrepresentation by Black and African American students and Hispanic and Latino students. These reporting agencies really only report gender as a binary, which I know is problematic. But I'll just note that with the data that they provide, we do see that there's also discrepancy, this kind of intersectional discrepancy between male and female students in these underrepresented groups. And this analogy of kind of there being a leaky pipeline where, female students kind of enter this pipeline, the female students are the water, I guess, in this analogy, the pipeline has holes in it that students leak out. And at the end of the pipeline, which presumably is a professorship or some professional career, there's a lot less water that has made it to the end of the pipe. Well, we see this to be kind of amplified for non-white women. So these numbers are even more extremely low when we look at other women who are not white women. And so I only mention this just to say that we are working as a group, a collective called Entomologists of Color, to try and do retention, recruitment, and advocacy to diversify the field of entomology. And I'll briefly mention one of the initiatives we had, which is that we are providing free memberships to all students of color to any entomological society, up to 3 per student in the world. There's like these forms on our website that you can just click on and sign up. We've fundraised, we have free memberships from some societies, plus we also fundraise. So please spread the word if you know anybody, grad student, undergrad student that wants free membership to academic societies, anyone, just let us know. So far, we've highlighted the students that have received these memberships on our social media sites. We've given over 300 away so far, as well as we organized a mentorship program for the last Entomological Society of America meeting. And we have a new grant that we just received that is in collaboration with a sociologist. So we plan to be sending out some surveys and questionnaires to various departments kind of across the United States that have entomology programs so that we could get at what's working, what's not working, and things that might be best practices and things that might need to be improved. So look out for that perhaps this summer. And with that, I would just note that the ultimate goal is this. This picture kind of summarizes what we want. These are my children when I took them to Guyana, and they have the same wonder that I had when I was a young student of color. I was interested. It's not that we're not interested. I was interested and I was passionate about entomology. I just didn't have the background, the family background, to know that this was something that I could do. But the goal is to try and make it so that entomology can be for everyone. And ultimately, we all would look at a box like this and be super excited about the diversity that we see, and it would spark a bunch of questions that would lead us to our work. So with that, I will just say thank you to my collaborators, my funders, my colleagues. Thank you to the people that I work with in my lab, and my museum specialist, Ruth Salas, and my former graduate students as well and postdocs. And if there's time, I'll take any questions. Thank you.
Thank you. What an amazing talk. Thank you so much. Applause. We have a bunch of questions coming in. Also, I love the Doctor Who background. Incredible. The first question that's coming in, as we should be. The first question that's coming in from Cheyenne is, with such a wide range of insect diversity, how did you choose to work with these groups? Was there something in particular that drew you to them?
Oh, that's a good question. Well, I'd be lying if I said I was drawn to termites and cockroaches. I wasn't, right? I was always drawn to dragonflies and damselflies. I spent a lot of time in Northern Ontario, kind of by a lake, and I would see them kind of landing. And I always thought they were kind of interesting. And they were the first group that I worked on as an undergraduate when someone kind of gave me an opportunity to do undergraduate research. But for termites and cockroaches, I kind of fell into them. I chose them for a term paper in grad school that ended up turning into a study on outgroup selection, which we published. And then I wrote an NSF postdoctoral fellowship on termites, and it was funded. And then I wrote my first grant that got funded for the NSF was on termites, and that's kind of how I stayed in the business. But now I love them. Now I can appreciate all sorts of things about them. But when I was first starting out, I wouldn't have said that they were my, oh yeah, I'm going to devote my life to that. Not, not really, but now I'm happy I am.
Right, and the second question that we have is from David Madison, and he says, "Hi Jessica, very nice talk, fascinating story about Margaret Collins. My question, as you know, there's a very large number of odonate fossils from the Mesozoic, most of them just wings. Have you or anyone done a tip dating analysis including those fossils? And if so, where do they end up in the trees? Scattered around as stem group lineages, or do the wings not have sufficient number of characters to place them confidently?"
I think it's the latter, really. So there was somebody in 1996, Jarzembowski, who did this. It wasn't really tip dating, but he scored them with morphology and reconstructed a phylogeny. And they were kind of all, it was an unusual phylogeny. And he and John Truman also did a paper around the same time that found that it really wasn't well supported. There's about 15 good wing vein characters that people routinely have been using since Tilliard, and there just doesn't seem to be enough signal. We have done some tip dating with them, and if you include them with molecular data, and if you include them, if you're able to score anything else from the compression fossil about their thorax or their tibia, sometimes you can actually reconstruct them in a location that you think would be feasible. But more often than not, I mean, Seth Bybee is someone at BYU. He did, for part of his PhD thesis, he again, it wasn't actually dating, but he scored morphology for fossils, and he included them. He found, some groups were easier to place in the topology than others. He has a graduate student right now named Robert Erickson, whose PhD thesis has been really trying to kind of expand what possible clues you can get from these fossils. So it's not just, wing fragments, but I think that especially with some of them just having kind of the post-nodal range. So there's a nodus, which is the middle part of the dragonfly wing. And a lot of them have either just like from the nodus to the tip or from the nodus to the base of the wing. And so you already are missing, some really important clues that are diagnostic for even family level kind of placement. So I think it's a little bit, it's tricky to use them for sure.
Sounds really difficult and hit or miss. We have some more questions. From Tall, we have one, Tall Levi, who asks, do you expect branching in the phylogenies to coincide with the period after major extinctions over the past 400 million years?
Do I expect branching to coincide with major extinctions?
Or maybe I think the question is after, like after a major extinction, do you expect there to be a burst of diversification?
Well, I mean, people have tested this. I mean, there's certainly friends. So there's this guy named Condamine who's done some work in France, who has done some tests on this. It doesn't seem like there's a signature across you would think that across the insect tree of life, you would see signatures of the last 5 kind of mass, or at least maybe not the last 5, the last 3 or 4 mass extinction events. But we don't actually see that with the data that we have. When Condamine did it, I think he was just using kind of Sanger sequencing that he had pieced together. I don't know if you had, better estimates of substitution rate, whether you actually would be able to see some type of correlation between extinction events and, kind of shifts in speciation rate. But from what I know, what's been done, I don't know that people have seen that. I would have, if I hadn't seen Francois' work, for example, or my postdoc Manpreet has done some work on this too. If I hadn't seen their work, I would have said for sure, I bet you're going to see these bursts. I mean, there is so much extinction that probably any of the sampling that we're doing for some of the groups of odonates, we're really just kind of sampling here, even though there's all this bushy part that we're missing. So maybe the substitution rates are useless, right? And so then we actually really can't kind of compare speciation rates across these taxa. But say that we are able to, I would have thought for sure that you would have seen it, but I don't think people have found that so far, from what papers I've read at least. They've had a lot of things to, I mean, so color is one thing that we think seems to be correlated with upticks in speciation rate. Shifts in habitat for dragonflies is something that we see seems to be correlated with speciation rate. Shifts in diet, that's something that seems to, you can kind of track that across the insect tree of life. There are some things that people have been able to see that seem to be related with kind of bursts, as well as these innovations like wings, pterygoda, social behavior. There are some things that seem to, be notable, things of note.
It looks like we have another question here from Kim Hobb. It says, thank you for your talk. Why do you think it is important to determine these phylogenetic trees? I know taxonomy can have a huge impact policy-wise for other species such as birds and mammals, for instance, in protections afforded by the Endangered Species Act. But since invertebrates tend to be ignored in conservation, why do the distinctions matter?
Well, I mean, it's such a good question because at some point they probably don't matter depending on the taxonomic level that you're interested in. Whether or not there are 3 suborders or 2 suborders within Odonata probably isn't going to affect even conservation. I'm interested in, my Nana would be interested if she read one of my papers. She'd say, oh, Jessie, that's really interesting. But other than that, it's probably not super interesting to people. But at the genus level or at the species level, sometimes so things like you mentioned can be really, really informative. So we often are interested in using those species level examples because they do impact conservation. We've done some work, for example, there's a genus called Syncordulia, which is found in South Africa. And it was really kind of going on the way to being vulnerable or listed as threatened by the IUCN, in part because it's very sensitive to riparian cover. So it lives in kind of freshwater streams, and when there's a lot of shade over the stream, populations were doing very poorly. Numbers were plummeting, and it was because in large part Australian acacia had been planted as an ornamental plant, and it was kind of taking over along the banks of these stream areas. So the South African government wanted to know, should they invest money in ripping out this Australian acacia, is it going to make a difference for Syncordulia? But in order to do that, they kind of wanted to know how many species they even had and what the kind of biogeography was of the taxa. If the taxa are actually all from Namibia originally, then why would they put the money into it? So we were able to sequence them and use molecular and morphological data to justify that there were these 4 species, and they had this, they had kind of originated in the Cape and had kind of radiated outward. And so then the government was able to use that, my colleagues in South Africa and the government were able to use that to justify kind of funding the removal of Australian acacia in these primary habitats, and the populations rebounded. So that would be a good example of when a phylogeny was meaningful. But again, that's like at the species level. Like I said, at the, whether or not gomphids and pedalurids or sister taxa, maybe it's not fundamental to the human condition. It doesn't maybe help the human condition in some way.
It's a dangling question we all must know. Gomphids and pedalurids, or gomphids and
Libelulites, yeah.
Libelulites.
It's the closest you can get to a time machine, really. Assuming like I said, I'm a Doctor Who person, but like assuming time travel is not possible, then the phylogeny is the next closest thing to kind of help us understand what happened 200 million years ago, 300 million years ago, and what have you.
Ooh, speaking of what happened in the past, transition, we have another question here, and it's going to be our last question before we kind of move to happy hour and a more informal question, just hang out. So Maria asks, is color something that you can see in the genetics? Can you predict color of species found as fossils?
Oh, well, so we aren't really able to get much genetic information from fossils. People have tried from some amber things, but it's largely been plagued with contamination, and it's not really feasible. But what you can see color from fossils, because some of the color that dragonflies and other insects have are what's called structural pigmentation. So there's kind of rugosities or bumps and ridges in the cuticle. The light reflects off of that, and our eyes perceive it, or other organisms perceive that as a color, often metallic color. So for some dragonflies and damselflies, for example, that are metallic green, you can, like, basically the cuticle, like, you can see those rugosities in the fossil, and when the light hits it, you see it as being metallic green. We can look at the genetics. There are genes that are associated with specific pigments. And then there's also, this is another thing that Seth Bivey's lab has worked on, and also Ryo Fukahashi in Japan has also worked on this, but you can actually look at the ability to perceive color, which is another part of the story. Having color is one thing, but then the ability to perceive color, since a lot of the coloration, especially for dragonflies and damselflies, a lot of the coloration is communication between males and females. So there are opsin pigments in the eyes that allow you to perceive different wavelengths. And some taxa have, A, B, and C, some have A and B, some have A and C, and what have you, and various combinations of opsins allow, perception of specific colors. So you can use that type of genetic information to make predictions about what colors should be available in the landscape if there's been selection for that opsin pigmentation in the eye.
Awesome. Wow. This is the first I have learned that you could see like a metallic green in a fossil that's however many years old. So that's really cool to know. Thank you so much. I think I want to just quickly say thank you on behalf of everybody here, on behalf of the EECB team and all of the departments associated. Just thank you so much for your talk, and thanks for coming and joining us.
Thanks for having me.
Yeah.
Applause.
Thank you.
And maybe Tal, you wanted to make a note before everyone's starting to slip out, but next week it seemed like who do we have up next?
Michael Cole. I've added that in the chat, and we're gonna have a debate about how wolves influence rivers in Yellowstone National Park. Between Michael Cole, Bob Beshta, and Luke Painter.
Wow.
I know. I think if we put this on BBC, we'll get a couple million people.